Welcome. Thanks for joining our satellite symposium tonight. As you see, the topic is, I think, new for a lot of us. The delicate balance of brain cholesterol and implications for developmental and epileptic encephalopathies. So my name is Dennis Dlugos. I direct the epilepsy program at Children's Hospital of Philadelphia. And I am very happy to share the podium tonight with Dr. Melissa Barker-Haliski and Dr. Nicole Hawkins, who are experts in brain cholesterol preclinical drug development. And I know I have learned a whole lot from working with them over the last few months putting this program together. And I hope you'll do the same tonight, and I'm confident you will. So this is our high-level agenda, some quick introductions, a little warm-up, and then a few talks, and then we have purposely dedicated a substantial amount of time for a panel discussion, and the audience is welcome to participate in our panel discussion, and then question and answers, because we realize this topic is new to many of you, and so the whole point is to be interactive once we get through the initial presentations. So credit is available for physicians, nurses, pharmacists through the typical organizations, and a quick way to access all of the information you need to claim the credit is to scan the QR code in the upper right-hand corner, and that will give you all of the information that you need. These are our disclosures, and note that all relevant financial relationships have been reviewed and mitigated by the France Foundation, which has coordinated the program tonight. And thanks to Takeda, the sponsor of the program, Takeda Pharmaceuticals, they provided an independent medical education grant that made this evening possible. Your phone will be important tonight as part of our interactive polling. So first, in case any of you are having trouble getting a cell phone signal, there is Wi-Fi that is available, and the details of the Wi-Fi are on the cards at your table, the name of the Wi-Fi network and the code. So Wi-Fi's there, and then once you have connected, there are two things that you need to do that are listed on the slide. First, in bold, send a text message to 22333. Then type a message that says TFF2 and hit send. And that should bring you into our interactive polling software. So I'll just give you a second or two to do all of that. All right, well, let's get warmed up with some pre-test questions. So if you haven't noticed, we're in Orlando, Florida. So the first question is, what were Mickey Mouse's first words ever spoken? And you see the choices there. Enter your choice with an A, B, C, or D. All right, we're at 20 responses and growing. All right, we've leveled off here. So, survey says, oh boy, with a large majority. And what was the correct? Oh, we're not going to tell you the correct answer yet, because this is pre-test, so you have to hang on if you want to know the answer. All right, next. You guys know the correct answer to this one already, so please let us know who you are. MD, DO, PhD, PA, etc. So choose your professional designation. All right. Thank you. Tell us your primary area, your primary clinical focus. Pediatric neurology, pediatric epileptology, adult neurology, adult epileptology, primary care, or something else. How many of your patients have DEEs? Now, we haven't formally defined it yet, but based on your knowledge right now of what a DEE is, how many of your patients have DEEs in a typical month? All right. I don't see the numbers rising. All right. How many, let's try this. How many of your patients have DEEs in a typical month? There we go. Thank you. All right. So that's very helpful for us to know that many of you are not clinicians. It's actually not surprising since this is cutting edge information. So that's very helpful to us as we craft our program and our discussions. So for those of you that are clinicians, what are the main challenges you face when treating and managing patients with DEEs? All right, yeah, well, so comorbidities, limited available therapies, and a little bit of all the other answers as well. All right, thank you for participating in our warm-up polling. There'll be more questions sprinkled throughout the talk, and then there will be questions at the end of the program. All right, so I will spend about 10 minutes trying to unpack what this phrase DEE means. It's relatively new and it is being used all the time. It is all over this meeting. So what is a DEE? So we'll go through some very basics about epilepsy and seizure classification, including we'll dive into the definition of DEEs, and you'll find out a key part about DEEs is can improving seizures or improving the EEG actually improve development? What do we know at this point about our ability to improve development by better treating seizures? A few comments about precision therapeutics, and then we'll move on. So the learning objectives are here. We'll start with unpacking the definition of a DEE. So this slide, in one slide, provides an overview of epilepsy, and it is a very useful slide taken from the ILAE classification organization system of epilepsy published in 2017. So, no surprise, epilepsy involves seizures that can be focal, generalized, or of unknown type. Epilepsy has one of 6 etiologies listed on the right, including unknown. Patients with epilepsy have comorbidities or associated conditions that are top to bottom, vast. A large majority of patients with epilepsy have a condition that is associated with their epilepsy. Just like seizures come in various forms, so does epilepsy. Your epilepsy can be exclusively focal or generalized or combined or also unknown. And then some patients have an epilepsy syndrome, which are specific types of epilepsy characterized by age of onset seizure types, sometimes EEG abnormalities, and typically some projection about prognosis. To dive a little bit deeper into the seizure part of epilepsy, this also from a 2017 ILAE, International League Against Epilepsy publication. This is the current organization of seizure types. There are focal onset seizures that may have a motor component, that is something stiffens or shakes, or may have a non-motor component, which means it is a seizure, but nothing stiffens or shakes. And awareness in focal onset seizures can be preserved, impaired, or impossible to know. And focal seizures may or may not progress to bilateral tonic-clonic seizures. Generalized seizure types, again, there are motor and non-motor seizures. With generalized seizures, the non-motor seizures are lumped under the term of absence, stop, stare, pause. And then a huge advance, certainly to pediatric neurologists in this classification system, is the third column, which means it is okay to do the most detailed history you can, do all the EEGs you can do, image, genetic testing, all of that, and you can't tell whether there are focal onset seizures or generalized onset seizures. That is no longer shameful. That is no longer being lazy. That is the reality. That unknown seizure onset is common, and the classification reflects that reality. So this slide is not rocket science. The goals for our patients include trying to identify what a seizure is and what it is not, identify triggers and then minimize the triggers if possible, to reduce seizures and minimize side effects as much as possible. The slogan is no seizures, no side effects, which is easier said than done, especially if no side effects really means no side effects, like none. Like I take a treatment every day, often twice a day, and I have 0 side effects. That is the goal, but I think a theme throughout the program is going to be tolerability side effects are key, and they are limiting steps with our current treatments, and they may be limiting steps with some of the novel treatments. So extra attention to tolerability side effects throughout the entire drug development process, I think, is very important. And then, of course, things that don't directly relate to seizures, like improved quality of life and increased independence. So one of the things I want you to think about is, after you hear about DEEs, and we unpack the complicated definition of a DEE, do those 3 words add anything to this slide? Maybe, but we're still trying to figure out what a DEE is in clinical practice and clinical trials, and does it advance the field beyond this very important starting point for clinical care with epilepsy? All right, so before we get to the DEE, let's take the last 2 letters, EE, epileptic encephalopathy. And these are all taken from the ILAE publication that introduced this term. So first thing about epileptic encephalopathies is they are rare. They are extremely rare. The notion is, prior to whatever happens with your seizures or your EEG, you have typical development. You are fine, completely fine. And then something happens, either a very high seizure burden or a very abnormal EEG, and you are not fine. The seizures or the EEG drags you down, but prior to that, A-OK. They can occur at any age, and treating the seizures or improving the EEG in this situation flips you back to being fine again. That's great when it happens. It is uncommon, but it is part of the DEE picture. So, an example. So, quick EEG primer. This is 17 seconds of a normal EEG in anybody who's like 8 years old or older. Turns out they're actually falling asleep during these 17 seconds. But this EEG is beautifully normal, as is the patient is typical. They're fine. But next slide are the next 17 seconds of their EEG. About halfway through the page, that normal drowsiness is replaced by bilateral generalized spike activity, epileptiform activity. And then it continues, and it continues, and it continues all night long. What is that? Well, it has a name. It has a new name as of 2022. It's called SWAS, spike wave activation with sleep. It is rare. It is very rare. It is particularly rare in patients with pre-existing typical development, no underlying developmental concerns, and no etiology. But it does happen out of the blue. And if you can normalize that EEG, sometimes easier said than done, they go back to being fine again. So that's a rare but dramatic example of an epileptic encephalopathy. The EEG drags you down, fix the EEG, you come back to your typical level of functioning. Now, extremely common are developmental encephalopathies, which means your development is different, delayed, atypical, and seizures are not really part of it. Or if they are rare. Your EEG is non-specific, nothing dramatically abnormal, which means there's nothing to fix. So developmental encephalopathies are extremely common, and they are often static, but they can go through periods of slowing or regression, which are unexplained by seizures or EEG. So this is extremely common, and one of the frustrating parts of it is there are no seizures to fix, there is no EEG abnormality to normalize. So then there's the DEE, which puts the two together. And DEEs are common, although not as common as developmental encephalopathies. So for a DEE, there is a pre-existing or coexisting developmental challenge, atypical development that may have an underlying cause, but may be unknown. And in addition, there are seizures, and there are often frequent seizures, and there are EEG abnormalities that may be profound EEG abnormalities. So you have two challenges. You have an underlying etiology, which may or may not be identified, which is impeding typical development, and on top of that, you have seizures and an abnormal EEG. The causes are vast. Acquired, extreme prematurity, stroke, head trauma, genetic abnormalities, or unknown. The big question with the DEE definition is, does the cognition or development of the patient improve if you control the seizures better and you normalize their abnormal EEG? That's the hope of the DEE concept, that effectively treating seizures or improving an EEG in some patients not only helps their seizures, but helps their cognition or their development. If you're going to do that, if we are going to do that, having safe and well-tolerated treatments are key. Because guess what? We can stop any seizure, and we can completely remove epileptiform activity from any EEG, no matter how abnormal. It's just a question of how long you want to be in the ICU and how many drugs you want in your pharmacologically induced coma. So the challenge is safe, tolerable medications to minimize seizures and improve EEG to the extent that's possible. So that's unpacking the DEE concept. So how do we do with this? How are we doing with developmentally, with improving development with treating seizures? So these are 5 examples here, and I'll go through them quickly because frankly, we're not doing that well. There's maybe 1.5 examples where treating seizures or EEG improves development. So neonatal seizures. This is a consensus statement that summarizes vast amounts of data that says treating newborn seizures, including EEG alone seizures, may be associated with improved developmental outcomes. And the level of agreement from this expert panel was moderate. So, okay, so maybe. The clearest example is an infant type of epilepsy called infantile spasms, begins in infancy, 6 to 18 months of age, comes with a profoundly abnormal EEG in many cases, which is highlighted here. This is EEG chaos. And the goal of treatment is to eliminate spasms and normalize the EEG. And we can do that sometimes. So when we do that, does development improve? Well, it turns out yes, but in certain patients. So this is older data from Israel, but it is still probably the most profound data about improving development based on treating seizures and an EEG. So these were 150 patients with infantile spasms that were treated a long time ago. The key is the 23% of them who had typical development, normal imaging, no cause found, no other seizure types, and a very typical pattern of that EEG chaos called hypsarrhythmia. Those patients received a lot of steroids, high-dose steroids for a long time, longer than we typically do now in the US. But the remarkable thing is, many years later, 6 to 21 years later, the 22 of the patients who walked in the door like this had 100% typical development. If treatment was delayed, the chance of typical development went down. So with no underlying etiology, a lot of aggressive treatment was associated with typical development. It's not class 1 randomized controlled data, but it's long-term follow-up data. Subsequent to that, there had been hints that maybe we can replicate that. UKISS was a trial that compared prednisone to vigabatrin in the treatment of infantile spasms, and in the group with no etiology, there was a hint that if you had hormonal, that is steroid treatment, your developmental score was improved over another treatment called vigabatrin, which raised hope that hormonal treatment can improve development, but that was not verified in a subsequent study that actually used combined hormonal treatment with vigabatrin. So the fact that early aggressive infantile spasms treatment improves development it may be true for a unique subset of patients with a lot of high-dose steroids for a long time, but it is not generally true. This is very recent data, lots of hope behind this study called the PREVENT trial. So tuberous sclerosis complex is typically diagnosed on prenatal ultrasound right now because they have cardiac abnormalities. A very high percentage of patients with tuberous sclerosis will go on to develop seizures, including many develop infantile spasms. So the magic behind this trial was to follow patients with tuberous sclerosis who did not yet have seizures. And at the first sign of an abnormal EEG, to randomize them to treatment with vigabatrin or placebo, and then see if development improved. And if you never had seizures, good for you, you didn't get treatment. And if you had seizures without having an abnormal EEG, you were also treated. Long study, lots of hope, and the bottom line was, with this design, it didn't affect development. Pre-treatment with vigabatrin did reduce spasms and delayed seizure onset, but did not improve development. One comment about precision therapeutics, so monogenic epilepsies. You know, this gene, that gene, ASO therapy, gene therapy, modify the RNA, the DNA, lots of potential, but the key is how early does it have to be given to not just treat seizures, but improve development? And we don't know. And there are many challenges to this, and we can discuss this in as much detail as you want in the, either in the panel discussion or in the Q&A session. So despite these challenges, we have treatments approved by the FDA for, I've listed here, 6 DEEs. These all went through the rigorous regulatory process, so there is a track record to get treatments approved for DEEs. The challenge is they treat seizures reasonably well, none of them perfectly, but the developmental gains are not necessarily coming as robustly as we'd like, so how can we make that better? So the key takeaways are the DEE definition is complex. It was not designed with clinical trials in mind, but it can be applied to clinical trials carefully. And our ability to improve development by reducing seizures is not great. With our current treatments, we have a lot of work to do. So we need new, safe, effective, well-tolerated treatments. And with that, I am very happy to hand off the microphone to Dr. Nicole Hawkins from Northwestern University School of Medicine. Thank you.
Oh, this makes me real tall. Go down here. I'm Nicole Hawkins, and I'm going to go over the importance of brain cholesterol and its role in neurologic health. The learning objective of my talk is recognizing the effect of brain cholesterol turnover in neurologic health. This is the quick outline of everything I will go over this evening. So what is cholesterol in the brain and its function? What are the metabolites? What happens when the system is dysregulated? And how could we use this as a potential therapeutic target? So first off, we're going to do a couple pre-test questions. So again, here's the information on what you need to use on your phone. You can text 22333, and then in the text line, TFF2, or you could also log into the below website. Good? Okay, so the first pre-test question is, which type of brain cell performs the majority of cholesterol turnover, and when impaired is hypothesized to lead to neurological disorders? Is it astrocytes, microglia, neuron, or oligodendrocytes? Oh. Yeah, it only has 18. You want me to try the next question? Okay. The second question is, during cholesterol metabolism in the brain, which of the following biomarkers is the primary metabolite that can be measured in humans using plasma or cerebrospinal fluid? That one's better. Yeah. All right. We're not giving out answers. All right, so cholesterol in the brain, maybe this is something that we're not all familiar with. So cholesterol is actually highly enriched in the brain. So while the brain is only approximately 10% of the total body mass, it contains 25% of total body cholesterol. We have two distinct pools of brain cholesterol. One that's metabolically stable, and this is located within the myelin sheath, and that's about 70 to 80%. The remaining 20 to 30% is located in the plasma and subcellular membranes of neurons and glial cells of the gray matter, and this is the pool that is metabolically active. So what are the functions of this brain cholesterol? So we have this, it has an essential structural component of the cellular membrane and in myelin. It's a precursor to steroid hormones and bile acid synthesis. It's a required component for the synapse and dendritic formation and axonal guidance. It's essential in neuronal physiology and development and also in adults. And cholesterol depletion in neurons impairs synaptic vesicle exocytosis, neuronal activity, and neurotransmission, and then dendritic spine synapse degeneration. So it plays several different functions. And then we have this animation.
Cholesterol acts as a precursor molecule for steroid hormones, including pregnenolone, progestins, testosterone, cortisol, aldosterone, and estrogens, which are essential for healthy human growth and development. Cholesterol is also abundant in the brain and required to maintain proper function. It is a critical component of cell membranes and the myelin sheath that covers nerve fibers. Humans obtain cholesterol from their diet, but most of it is synthesized in the liver. However, the blood-brain barrier prevents peripheral cholesterol from entering the brain, so the brain synthesizes cholesterol de novo. The brain consists of various cell types, including neurons, astrocytes, oligodendrocytes, and microglia, each having unique cholesterol requirements. Cholesterol turnover and metabolism is essential to brain health, with a closely maintained balance between biosynthesis and elimination. CYP46A1, cholesterol 24-hydroxylase, converts cholesterol into 24S-hydroxycholesterol, also known as cerebrosterol. This compound passes through the blood-brain barrier and undergoes further metabolism in the liver, where it is transformed into bile acids. 24-OHC has been extensively studied as a biomarker for brain metabolic activity, particularly in neurological disorders. It plays a crucial role in modulating various receptors and ion channels, including the NMDA receptor. The modulation of NMDA receptors in the brain is important for synaptic plasticity, learning, and memory. Additionally, 24-OHC acts as a positive allosteric modulator of NMDA receptors. However, overstimulation of NMDARs can lead to excitotoxicity and neuronal cell death, seen in conditions like status epilepticus and hyperalgesia. CYP46A1 is thought to contribute to the pathogenesis of certain neurological disorders, including status epilepticus. Activity of this hydroxylase can increase neuronal oxidative stress via the glutamate-mediated excitotoxic pathway, leading to seizures. Results from various mouse-based behavioral learning assays suggest that CYP46A1 plays a role in higher-order brain function. Excessive glutamate receptor stimulation can also lead to decreased cholesterol levels due to CYP46A1 activity. Interestingly, in vitro CYP46A1 can be activated by glutamate, suggesting a reciprocal relationship. Increased CYP46A1 activity boosts excitatory neurotransmission, which in turn increases CYP46A1 activity. Therefore, CYP46A1 inhibition is hypothesized to be useful in managing conditions that lead to excessive glutamate activity like schizophrenia, status epilepticus, and hyperalgesia.
All right, so now we're going to unpack some of that animation. So cholesterol synthesis within the brain. So as mentioned, brain cholesterol is derived from within the CNS. The plasma lipoproteins cannot cross the blood-brain barrier, and they also cannot leave the CNS through the BBB. This biosynthesis entails over 20 different enzymes that convert acetate into cholesterol. And there are multiple different pathways that neurons and astrocytes use to generate cholesterol. Within the brain, astrocytes appear to be the major supplier of neuronal cholesterol in maturity, and these are being delivered to neurons via ApoE. On the other hand, excitatory neurons express the machinery for cholesterol synthesis and homeostasis. And these could be located within the cortex and hippocampus and pyramidal cells, and also in Purkinje cells in the cerebellum. So we're going to get into a little bit of alphabet soup, as I always say. So brain cholesterol and 24-OHC. So 24-OHC is the major metabolite of cholesterol. This is a side chain oxidation product. You'll see it as 24-SHC, 24-HC, or 24-OHC. And this is the dominant mechanism of cholesterol catabolism in the brain. So 24-HC is produced by the cytochrome P450 CYP46A1. The gene name is CH24H, so those you could see also interchangeably. CYP46A1 accounts for approximately 75 to 85% of brain cholesterol turnover. This expression of CH24H begins early in life, and it reaches steady state levels at about 1 year of age. This rate of cholesterol catabolism is closely coupled with the rate of synthesis, and 24HC is commonly referred to as a cerebral sterol. So lots of names. So if we look at this pathway with CYP46A1 and 24HC, the major excretion pathway, as demonstrated in the animation, is through bile acid synthesis. And this process is strictly neuronal. CYP46A1 expression has been identified in the hippocampus principal cells, and it's been identified strictly in the soma and dendrites. It hasn't been identified within the axons and presynaptic terminals. They've also found little expression of CYP46A1 in glial cells, so this is indicating to us that major cholesterol turnover takes place in the neurons, not the astrocytes. So 24HC influences various biological functions. So CYP46A1 is activated by glutamate. 24HC is a positive allosteric modulator, or PAM, of NMDARs. And so this is going to enhance the calcium currents through the NMDARs. There was a knockout mouse of CYP46A1, or CH24H, and that demonstrated a significant reduction in 24HC levels by 60 to 80%, and the remaining peripheral cholesterol was normal, and there was a 40 to 60% reduction in this brain cholesterol synthesis. So what is this role in neurotransmission? So in vivo and in vitro experiments have revealed this reciprocal relationship between 46A1 activity and neurotransmission. And you can see we have this kind of circle that just repeats and repeats. So we have this excitatory neurotransmission that's increasing the CYP46A1 activity, and then that increased CYP46A1 activity is enhancing neurotransmission. So you can see how this could be important in implications with your NMDARs with hypo and hyperfunction. So what happens when we have dysregulation in this cholesterol pathway? So we have something called excitatory amino acid transporters, or EATs, and it's a major glutamate transporter in the central nervous system. EATs are associated with cholesterol-rich lipid rafts, and also reduction of cholesterol has revealed reduction of cholesterol revealed reduced EAT uptake activity. So if the EATs aren't taking up the glutamate, then you're going to have more glutamate within the synapse. That can lead to increased neuronal excitation. Furthermore, when CYP46A1 is seen to be decreased in neurons, we see it increase in astrocytes. And so then that's where we have that extrasynaptic glutamate. So when do we see altered 24HC levels in neurological disease? And yes, we do. This 24HC is a clinically monitorable biomarker. You can do it in plasma or CSF. This enzyme activity can be, it's easily analyzed, and because we know it's majority brain expressed, we would see what was happening. So you can see here what diseases are involved with altered 24HC. So schizophrenia, autism spectrum, Huntington's, Alzheimer's, just to name a few. And I'll go over how these levels have changed in these specific neurological diseases. So in Alzheimer's disease, they identified varying changes in plasma 24 expression based on the disease stage. So in early Alzheimer's, 24HC was mildly reduced, while in later stage Alzheimer's disease, they saw 40% reduction in 24HC. Huntington's disease, there was reduced plasma 24HC at all stages, but was completely normal in pre-Huntington's patients. For MS, there was 24HC reduced in primary progressive and older relapse remitting patients. And Parkinson's, the higher 24HC was inversely associated with risk of the disease. And lastly, autism was identified as having increased 24HC levels. So can we use CYP46A1 as a potential therapeutic target? So going after all of the previous data where you can say that a CYP46A1 activation could be beneficial for perhaps Alzheimer's disease and Huntington's disease, where your pathology is this reduced brain levels of CYP46A1. Therefore, you're going to be increasing the NMDAR function. If you can inhibit CYP46A1, you could have a therapeutic value in pathologies that include excitatory glutamate, such as epilepsy and hyperalgesia, schizophrenia. So this would be resulting in CYP inhibition would be decrease your NMDAR function. So using this, when we saw local overexpression of CYP46A1 by viral vector, that this was beneficial in Alzheimer's disease, ALS, and Huntington's disease mouse models. And activation was beneficial in this hyponeuronal function. In diseases with excessive glutamatergic excitation, including epilepsy, could we inhibit CYP46A1 and this be beneficial to patients? Does inhibiting this negatively modulate NMDRs and improve EAT uptake? So this is a very simplified schematic of what we're hypothesizing here, is that if you block the breakdown of cholesterol into 24-HC, you could potentially restore and maintain cholesterol homeostasis in hyperexcited regions of the brain. You could also decrease excitotoxicity from NMDARs and decrease extracellular glutamate, therefore potentially decreasing seizure susceptibility. So the key takeaways from this portion is that the brain endogenously synthesizes cholesterol from acetate. Cholesterol homeostasis is critical in the regulation of neurotransmission, membrane potentials, and synaptic vesicles as examples. Brain cholesterol is catabolized by a brain-specific enzyme, CYP46A1, also CH24H. This major metabolite, 24-HC, can be excreted through the blood-brain barrier and serve as a biomarker. 24-HC levels influence NMDAR activity and also an inflammatory response. Elevated 24-HC can create a hyperexcitable cycle through NMDARs and leading to glutamatergic overstimulation. And with that, I'm going to hand it off to Dr. Haliski.
Thank you, Dr. Hawkins. That was super to hear from. So I'm Melissa Barker-Haliski. I'm a research associate professor at the University of Washington, and I hope to educate you today about brain cholesterol dysregulation and the pathophysiology of developmental and epileptic encephalopathies. So my learning objectives are to evaluate the impact of dysregulated cholesterol in the brain and the implications for developmental and epileptic encephalopathies, or DEEs. I hope that we can assess the evolving strategies to integrate preclinical DEE models for novel therapy discovery for epilepsy. And finally, differentiate the preclinical profile of a brain-specific cholesterol catabolism in rodent seizure and epilepsy models. So here is the outline for the talk that I have prepared for you today. First, we're going to learn a little bit more about developmental and epileptic encephalopathies. We'll talk a little bit further, deeper dive into brain cholesterol dysregulation in some neurological conditions and how inhibition of brain cholesterol may be involved in managing those conditions. And finally, the key takeaways. So, whoops. Okay, more pre-test questions. Can you please help indicate for me, if you've got your phone, hopefully you still remember the text or URL, but which of the following is an advantage of preclinical models of DEEs? Okay. going to move on to the next question because we're going to. So the next question is, blocking brain cholesterol catabolism in an Alzheimer's disease mouse model reduced excitability and release of which neurotransmitter? GABA, glutamate, dopamine, or serotonin? Okay. Pre-test question number 5. Inhibition of which enzyme with soticlestat is thought to improve seizure outcomes? CYP46A1, ApoE, SCN1A, or TRPV1? Good. And pretest question number 6. Sustained status epilepticus has which of the following effects on cholesterol metabolism? An increase in 24-OHC levels, a decrease in free cholesterol levels, an increase in serotonin levels, or a decrease in ApoE levels? All right. We are all here for the American Epilepsy Society meeting, and we hopefully have seen this slide a few times. But we all should appreciate that epilepsy can affect anyone at any age, but there are particular age groups that are at higher risk, and this includes the very young or the very old. In younger individuals, the highest incidence of epilepsy presentation occurs within about the first 3 years of life, and you've got a nice summary of within the months of development when the breakdown is for the most frequent age of presentation, and it tends to happen very frequently within even a year of life. This is a quick breakdown of some of the classical epilepsy syndromes that correspond to DEEs, and it helps you see both their clinical naming convention, but also what kind of features of epilepsy or what kind of seizure phenotypes you might expect to see. The ones that you probably will want to key in on for the purposes of today's talk and the discussion, the panel, are Dravet syndrome, where you have a normal child with a prolonged febrile or afebrile seizure with either focal and tonic-clonic seizures at onset, and that typically can present as early as 6 months of age. Now, the causes of DEEs, as Dr. Dlugos already alluded to, tend to be varied in nature, but we tend to see breakdown within the specific epilepsy syndromes, whether they're genetic structural, or genetic mutations alone. You can see specific breakdowns across the epilepsy syndromes, and I wanted to key in for you kind of in the middle of the graph with the Dravet syndrome population, where we see a lot of genetic components associated with the disease, whereas in infantile spasms, you can see structural modifications as well as genetic variants that lead to structural malformations that can cause the condition, as well as genetic or structural defects as well. Other issues that can contribute to DEEs include metabolic conditions, or as Dr. Dlugos already mentioned, trauma at birth, including hypoxia. So they're varied in nature, but this information as far as what causes these DEEs is really important to us as preclinical scientists as we begin to develop and implement our preclinical models. And so I'm a basic scientist. I like to think about preclinical models of epilepsy and how we can use them in the most appropriate manner to address a clinical question at the bench. So there are many preclinical models of developmental and epileptic encephalopathies, and they have some benefits, but they also have some problems associated with them. The benefits, as a basic scientist, to think about is that these transgenic animals have a high face and construct validity. Well, what does that mean? That means that the seizure presentation in the mouse model often replicates what is seen clinically, and we can introduce a genetic mutation to evoke that seizure presentation. That allows us as basic scientists to do really powerful mechanistic studies where we can say, okay, what's the role of this particular mutation from birth, or how, if we modify this particular target at a specific time point, how does that affect the trajectory of the animal's development? And then we can have conversations with our clinical colleagues about what that might mean going forward. This also presents to us a very powerful precision medicine opportunity. If we know the monogenic causes of the disease, we can then say, this particular mutation is very poorly tolerated in our mouse lines in response to a particular drug. Maybe we should move that forward in the future. And we can also modify when the expression occurs. We can have developmental expression from birth, or we can have conditional modifications that we can turn on or off the mutation to see what happens when it is or is not implemented. So that's great. That's really powerful. But there are also side effects or problems associated with these DEE models. And one of the most problematic is the high early life mortality that limits their utility for really powerful, statistically rigorous, high throughput studies that are important to assess safety and tolerability in a preclinical setting. That also introduces a very high cost or a barrier to implementation in a preclinical laboratory, as well as oftentimes the mouse model that's available at Northwestern maybe is different than the mouse model that's available at the University of Washington. And so there's limited investigator experience with those particular mouse lines, as well as most often the models are evoked in mouse lines, although that is changing in recent years. And all of those problems culminate to make these models very infrequently used for anti-seizure medicine or ASM discovery practice. And so why this is changing and why this is kind of an interesting point in drug discovery and drug development lifetime is Dravet syndrome has actually now provided us with a really unique and almost disruptive discovery opportunity for the 21st century. For the last 50 or so years, anti-seizure medicines have been brought to market on using wild-type, neurologically intact male rodents. But this particular model is now clearly defined to have be associated with a rare syndrome with a very clearly defined genetic cause. It can be associated with early life seizures that we also see in our preclinical models. The human patients are highly drug resistant, and we can also see that preclinically. We can see neuropathology. I've got a beautiful immunohistochemistry staining here of a wild type animal WT on the left and a Dravet syndrome mouse on the right, and you can see a high amount of reactive gliosis in the brains of those DS mice. That also corresponds to a high amount of comorbidities and cognitive delay in people, but we see that same feature in animal models. And the animal models, just as in human patients, have a high SUDEP risk. And so these Dravet syndrome models have been actually very recently implemented to be useful for anti-seizure medicine discovery. And so it provides us with a tool in which to say, okay, if a compound is effective in a DS model, maybe there's opportunity and hope for it to move to clinic. And why Dravet syndrome is particularly interesting from a basic science perspective is that syndrome-specific models, such as those that are modeled with DEE models, can maybe help us get at a bigger problem that's facing the epilepsy community as a whole, is that drug-resistant epilepsy is something that many of us think about when we go to work each day. But the underlying causes of drug-resistant epilepsy are very complex. And this is a great summary of the many kind of hypothesized mechanisms that contribute to drug-resistant epilepsy. You can see things like changes in the blood-brain barrier transporters. You can see changes in the molecular target. But not any one of these can fully explain the causes of drug-resistant epilepsy. And so we have to use that information to ask clinically relevant questions, but without doing that experimental investigation in a clinical setting, because it's really unethical to set people up for drug resistance. And so the syndrome-specific models offer us a way to uncover the factors that lead to drug resistance to then identify new therapeutic strategies. So this brings me into the discussion about DEEs and cholesterol. And so we already heard very nicely from Dr. Hawkins that disruption of brain cholesterol occurs in many different neurological and neurodegenerative disorders where excitotoxicity and seizures or hyperexcitability may play a significant role. Brain cholesterol accumulation, as illustrated in this figure of the post-squalene synthesis pathway for cholesterol, can accumulate, and the CYP46A downregulation can actually evoke excitotoxic effects and lead to neuronal death. And so you can see the pathway of cholesterol. It's maybe easier to see for you than for me. But ultimately, it's packaged in these neuronal endosomes, and that's where the majority of cholesterol metabolism takes place, is within those neurons. Now, dysregulation of brain cholesterol has been implicated in a variety of neurological diseases. So you can see two separate pathways here where you've got cholesterol going into the ApoE pathway, where it can contribute to synaptic plasticity and peripheral nerve regeneration. But the metabolic dysregulation pathway at the bottom shows you how cholesterol dysregulation may be involved in diseases like neurodegenerative diseases like Huntington's disease, Parkinson's disease, or Alzheimer's disease, or in neurological disorders like epilepsy. And so some of the really interesting evidence about how blocking brain cholesterol catabolism may play a role in hyperexcitability actually came from studies in an Alzheimer's disease mouse model. So this is a study, the first panel, the Kaplan-Meier curve on your left, was a study looking at amyloid precursor protein, presenilin-1 overexpressing mice, which is a model of Alzheimer's disease. And this study looked at the effect of it. And these mice are known to have spontaneous recurrent seizures. They're known to have mortality as a result of those seizures beginning at around 4 months of age. So the study looked at how administration of a specific CYP46A1 inhibitor, soticlestat, administration of this compound, whether or not it could block or change the trajectory of disease. And what's fascinating to see is that animals that are in the control group that were untreated show that typical mortality presentation. But if you administer soticlestat chronically beginning at 7 weeks of age, you don't see that same mortality. And so if you then dive deeper into why that might be occurring, well, if you take slices from these animals if you infuse potassium chloride into the brains of these animals and look at the extracellular glutamate dumping in that panel labeled A in the middle, you can see that there's the APP-PS1 mice in response to extracellular potassium chloride infusion show a huge increase in glutamate release that's not observed in the wild-type animals. However, in the next panel, C and D, what you can see is that those same APP-PS1 mice that are administered soticlestat, a compound that blocks the CYP46A1 activity, it will actually prevent that extracellular glutamate release, that spillover that happens. And you can see that quantified in the graph, the bar graph on the far right in panel D. And so this suggests that blocking brain cholesterol catabolism may play a role in blunting glutamate release and neuronal hyperexcitability. And if you carry that into some studies with status epilepticus, this hypothesis further plays out in that there's a significant dysfunction and disturbances of brain cholesterol metabolism that, as a result of status epilepticus, may lead to the excitotoxicity and neurodegeneration that's consistent with status epilepticus. Tracking the expression of this brain target, you can see that hours to days after status epilepticus insult, which is again a severe seizure emergency, there is a significant decrease in 24-OHC levels. But then, subsequent days after that onset, the free cholesterol levels start to go up. And so you see that second phase of the red line in the graph. And then as that time goes on, the sustained status epilepticus leads to neuronal death, which induces increased levels of 24-OHC. And as we heard about from Dr. Hawkins, that's a really useful plasma biomarker for brain disease. One other thing I wanted to highlight for us all today is just that the role of cholesterol catabolism and metabolism. It has also been implicated in learning and memory. I'm not going to dive into that too much for the purposes of this talk, but it may be relevant to future studies in a mechanistic setting. What is interesting as a basic scientist is that we have really nice evidence to demonstrate that blocking brain cholesterol catabolism can actually reduce seizure occurrence in two kind of more traditional temporal lobe epilepsy screening models, or models that are characterized or more useful in epilepsy space, not an Alzheimer's disease mouse model. First of all, if we look at the intra-amygdala kindling model, which is just a local infusion of canic acid that leads to epileptiform activity and spontaneous recurrent seizures weeks after the neurological insult, you can see that in that green box, the first panel, you see a significant reduction in the number of spontaneous recurrent seizures over a 2-week monitoring period in animals that are treated with soticlestat, the compound that blocks the CYP46A activity. However, it's important to note that even though the number of seizures were blunted in that particular treatment group, the average seizure duration of animals that were treated with that compound didn't change. The animals still had seizures, they just didn't have as many. Interestingly, in some other studies using a viral infection-induced seizure model, we also saw that animals normally that have this viral infection show a high incidence of evoked seizures during an acute infection period. But if we administer soticlestat during the treatment period, or during the infection period, you can actually blunt the number of induced or evoked seizures that occur. And this also carries significant effects on the generalized seizure presentation, or the racine stage 3 to 5 seizures, suggesting that two models of evoked seizures or spontaneous recurrent seizures do also demonstrate sensitivity to soticlestat administration. But thinking about DEEs, which is why we're all here, we also want to look at a more syndrome-specific model of Dravet syndrome. And what's interesting to think about is that in this Dravet syndrome mouse model, where the animals have a critical period in which they have spontaneous seizures, they have a reduced seizure threshold, and they can be used to screen for investigational therapies. When soticlestat was administered for a brief treatment period, the animals that were subjected to a hyperthermia-induced seizure threshold test, so just looking at how sensitive an animal was to having an evoked seizure, animals that received soticlestat actually showed an increased threshold to an evoked seizure. So they were more able to withstand the thermal induction method versus vehicle-treated animals that also had the Dravet syndrome mutation, suggesting that soticlestat may have an anticonvulsant, or at least ability to raise seizure threshold. But as you think about treating a developmental or think about a developmental condition, raising seizure threshold is great, but how does blocking brain cholesterol catabolism affect long-term outcomes? And so the next really interesting piece of evidence comes from a chronic administration study in the same type of Dravet syndrome mouse slide, where the animals are known to have a high degree of mortality beginning at around day 21 after birth. And you can see that in the two solid, or the solid black and the dashed black line, the two lines down at the bottom. Those are normal Dravet syndrome mice, both male and female, have about a 25 to 50% mortality rate by up to day 40 post birth. However, animals that had chronic administration of soticlestat in their chow showed significant improvements in mortality. There was none, suggesting that not only does this effect on brain cholesterol catabolism have improvements in seizure threshold, it can potentially also have a disease-modifying effect in a Dravet syndrome mouse model. I have another animation. Do I have to hit start? Maybe.
Alterations in brain cholesterol homeostasis is increasingly being recognized as a potential contributor to neuronal excitotoxicity, a phenomenon characterized by excessive exposure to the neurotransmitter glutamate or the overstimulation of its NMDA receptors. This, in turn, can lead to excitotoxicity causing neuronal injury or death. Excitotoxicity is the hallmark of various neurological conditions, including Alzheimer's disease, Huntington disease, and status epilepticus. Seizures cause excessive glutamate release, which in turn stimulates NMDA receptors, triggering substantial influxes of calcium ions into the cell. This cascade of events ultimately results in widespread neuronal death, partially attributed to the presence of inflammatory proteins. Following an initial seizure, a latent period may extend over several years, during which synaptic plasticity and the reorganization of neuronal networks undergo changes, potentially giving rise to cognitive alterations. The reorganization of these neuronal networks makes them more susceptible to excitatory signals, ultimately paving the way for the development of spontaneous and recurrent seizure episodes. Proposed temporal changes in free cholesterol and 24-OHC levels following status epilepticus can be categorized into 3 phases. In the initial hours and days post-SE onset, 24-OHC levels decrease, likely linked to transient CYP46A1 inhibition, while free cholesterol levels remain normal. Several days after status epilepticus initiation, free cholesterol levels rise due to the absence of a negative feedback mechanism initiated by 24-OHC on cholesterol synthesis enzymes, and 24-OHC levels return to normal. In cases of prolonged status epilepticus, patients experience neuronal death, leading to an increase in 24-OHC release. Studies have begun to examine the impact of blocking brain cholesterol catabolism as a therapeutic target for patients with developmental and epileptic encephalopathies, or DEEs. DEEs are characterized by severe seizures that are often drug-resistant. These patients also have encephalopathy, which can indicate significant developmental delays and loss of skills learned during development, such as talking or walking. Examples of DEEs include Dravet syndrome and Lennox-Gastaut syndrome. In a mouse model of Dravet syndrome, researchers demonstrated that inhibiting brain cholesterol catabolism via CYP46A1 signaling using soticlestat reduced susceptibility to seizures. Further, soticlestat treatment was shown to reduce the seizure burden and improve survival in Dravet syndrome mice. Inhibition of brain cholesterol catabolism is currently being investigated in clinical trials for DEEs. The Phase 2 clinical trial ELECTRA examined the use of soticlestat in patients with Dravet or Lennox-Gastaut syndrome aged 2 to 17 years old. The results revealed a statistically significant reduction in seizures from baseline compared with placebo in Dravet syndrome. However, patients with Lennox-Gastaut syndrome did not show statistically significant reductions. It is crucial to ascertain whether cholesterol accumulation resulting from the inhibition of cholesterol catabolism via CYP46A1 leads to neuronal death in humans. This is of particular significance, considering that inhibiting this enzyme has been shown to induce epileptic behavior, seizures, and neuronal loss in mouse models. Additional research is still underway to determine the safety and efficacy of this therapy in humans.
So the last thing I wanted to highlight today was kind of just a bigger picture. How do these DEE models and how does this activity with soticlestat in the intraamygdala kainate model, the Tyler's virus model of infection-induced epilepsy, and the Dravet syndrome model stack up against the anticonvulsant screening potential of many of the more traditional seizure models that are out there. And this includes mouse models like the maximal electric shock test, the subcutaneous pentylene tetrazole test, or the 6 Hertz test in mice at 2 different stimulation intensities. And what I want to draw your attention to is how these quote-unquote traditional screening models correlate to clinical syndromes and whether or not they have been found to have predictive validity for clinical efficacy. So the MES test, the sub-QPTZ test, those are 2 models that have really formed the backbone of screening for most of the FDA-approved anti-seizure medications that are on the market today, and have shown high predictive validity. Well, when soticlestat was administered to mice in these particular assays, it did not show any activity. And so it may suggest that a syndrome-specific model is a more appropriate way to ascertain the potential for novel therapeutic mechanisms to really be impactful for DEEs. And so the key takeaways that I want to leave you with today are how we can use this to impact clinical care and practice. DEEs represent a novel precision medicine strategy for 21st century epilepsy therapy discovery. These seizures in these patients are often treatment resistant, and we can oftentimes identify genetic variants that can be useful to inform treatment selection. Brain cholesterol biosynthesis is demonstrated to be dysregulated in many neurological conditions and neurodegenerative disorders. And 24-OHC is found in human plasma that is of cerebral origin, making a very useful clinical biomarker. So brain cholesterol biosynthesis in epilepsy is a novel therapeutic target, and it's effective in numerous preclinical drug-resistant chronic seizure models, but it was inactive in the more quote-unquote traditional and predictive screening models that we use for many therapies on the market today. And so with that, I'm going to turn it back over to my colleague, Dr. Dlugos. Thanks.
All right, thank you very much. So we're going to go back to the clinical realm for a bit before we have a discussion. So there are many new pathways, new mechanisms being explored for treatments of DEEs. Certainly brain cholesterol is one of them, but there are others, and we're going to do a quick review of what's going on in this field. So we'll look at the different pathways that are being investigated, and I do want to try to emphasize side effects of these pathways, because no matter what the treatment is, it will work in some patients and it will typically not work in some patients, and that is very hard to predict. What is easier to anticipate are side effects. A drug that's unsafe won't make it to market, hopefully, but tolerability side effects are, I think, a little more consistent between patients. And what do these new mechanisms tell us about potential tolerability side effects or lack thereof in humans? So here's the outline. But first, more questions. So what is your top priority when treating and managing patients with DEEs, in addition to limiting seizures? So assume you want to control seizures. What else is your top priority? And you see the choices there. Identifying seizures and triggers, improving patient recovery, following a seizure, managing side effects, addressing developmental concerns, or something else. All right. All right, addressing developmental delays, regression, and treatment-limiting side effects. What approach do you recommend most often to treat and manage patients with drug-resistant seizures? Surgery, neurostimulation, dietary therapy, immunotherapy, or other? And survey says, all right, neurostim and other, and surgery. So other would have included medications, traditional, novel, whatever. All right, those are helpful answers to set up some of the discussion to follow. Okay, so I showed this slide before. These are 6 DEEs that have FDA-approved treatments. And what I want to highlight is tuberous sclerosis complex can be very specifically diagnosed. clinically and typically genetically. There's not a lot of mystery in whether a patient has tuberous sclerosis complex or not. Dravet syndrome typically has a very clear phenotype with about 80% of patients having genetic confirmation. Also rather clear. CDKL5 deficiency, typical phenotype genetic abnormalities. Rett syndrome, typical phenotype, some genetic variability, although a most common genetic disorder. But then the two outliers, Lennox-Gastaut syndrome, there's no typical anything with Lennox-Gastaut. And then infantile spasms, also a wide variety in terms of etiology. The only thing that is consistent with Lennox-Gastaut and infantile spasms is an EEG abnormality that doesn't even necessarily persist for very long. It may be there and then it will go away. So why am I emphasizing this? So, by far, the DEE with the most approved treatments, and also, I think, the most clinical trials in progress remains Lennox-Gastaut syndrome, which is 1,000 different things. Yet, it is a common pathway to gaining regulatory approval for seizure medications. So this summarizes the recently revised criteria for Lennox-Gastaut syndrome, which is there's really nothing new here. It's a mixture of multiple seizure types, which typically includes tonic seizures, that typical EEG abnormality, and I'll show you examples, and then developmental differences. So it means so many different things. If you want to diagnose Lennox-Gastaut, and in particular, if you want to enroll a patient with Lennox-Gastaut in a clinical trial, what you need to show, for better or worse, is either this, which is an EEG snapshot of the typical EEG pattern of Lennox-Gastaut called slow spike and wave. Each of those vertical lines is a second, and there are roughly 2 spike wave complexes in each second. That is slow spike and wave, which is there sometimes forever and ever in Lennox-Gastaut patients, and other times is there only briefly and goes away. It is more commonly seen in pediatric patients than adult patients. We don't know really where it comes from or why 1,000 different etiologies tend to have this EEG pattern at times during the course of the disease, but it's extremely important in the clinical and regulatory diagnosis. The other EEG pattern is this, which is called generalized paroxysmal fast activity, which is just a brief burst of fast spikes. Incredibly nonspecific, can be there, can go away, can be seen in kids, adults, a little, a lot, and also a final common pathway of 1,000 different things that is not well understood. So what is Lennox-Gastaut mechanistically? Nobody has any idea. What is it etiologically? 1,000 different things. The honest role of Lennox-Gastaut is it is a pathway for regulatory approvals, which is not a bad thing. Patients with Lennox-Gastaut and other types benefit from that. But an issue in the DEE field is, have the regulatory definitions been too limited? So a broader DEE definition may be appropriate for future DEE trials. Lennox-Gastaut is an example, I think, of false precision. We don't really understand what it's telling us, and it is so heterogeneous, and it leaves out lots of patients. So perhaps another way to think of DEEs more broadly would be criteria like this. So the seizures do typically start early in life, so seizure onset 5 years or younger, a history of developmental delay, a mixture of seizure types, either focal and generalized seizure types or multiple generalized seizure types. The EEG background should be atypical, that is slow or disorganized, but the slow spike wave, the GPFA, it's there, it's not there. Is that really that critical for such a heterogeneous type of epilepsy? And then very important, patients with DEE do not fit into another category of patients, which are patients with typical development called idiopathic generalized epilepsy, but who have persistent seizures. Now that's maybe 20% of patients with what we call IGE have unmet needs in that they have persistent tonic-clonic seizures, myoclonic seizures, but they don't have developmental differences. They have unmet needs, but they are not DEE patients, I think, by clinical, and if DEE is ever endorsed by regulatory agencies, that's a different group of patients. So broaden the DEE definition, but not so broad that it means every patient with poorly controlled seizures, because that won't fly with regulatory agencies if the DEE definition is stretched too broadly. So there is a lot going on in the DEE space, and a lot of it is represented heavily in this meeting. So this is going to be a quick flyby of some of the novel therapeutic strategies that are being utilized to address DEEs. And these are vastly different strategies. So the first two involve neurostimulation, and remember when we did the polling, neurostimulation was high on the list of treatment options for patients with treatment-resistant DEEs. So there are two strategies that have some data to support their use, although no neurostimulation device has a regulatory approval for use in any DEE at this point. The first approach involves deep brain stimulation of electrodes in the bilateral thalamus and a particular nucleus of the thalamus called the centromedian nucleus. So this picture, imagine you're looking, this is a coronal section, so you're looking straight on at the patient, and the yellow is the centromedian nucleus of the thalamus. These are the ventricles. So a fairly small nucleus, and this is data from a paper, study done in Australia, where bilateral deep brain stimulating electrodes were placed in the centromedian nucleus in patients with Lennox-Gastaut syndrome, and it was not a regulatory trial. The outcome was interesting. Electrographic seizures went down statistically in a statistically significant way. Diary seizures, short-term, did not go down in a statistically significant way, but over the long term, diary seizures did go down. So it was a mixed outcome, but it was intriguing that electrographic seizures were reduced with deep brain stimulation. Now, this type of deep brain stimulation is autopilot. There's nothing responsive here. Nothing is sensing a seizure and then stopping it. This is chronic stimulation that can be adjusted within limits. And it at least hinted in this early Australian study of a reduction in seizures in patients with Lennox-Gastaut syndrome. So what's the magic behind the centromedian nucleus? Well, let's just say it's connected to almost everything in the brain. Old-fashioned neuroanatomy studies suggest that the centromedian nucleus has inputs from the brainstem, in particularly the midbrain, the basal ganglia, and it projects to the frontal lobes and the parietal lobes. So that's a lot of seizure susceptibility territory there. The other deep brain stimulation strategy involves responsive neurostimulation. So this is a little more complicated than the deep brain stimulation electrodes, and this is a study that is underway currently. It's literally enrolling. And these patients, so this is the skull, which has been exposed, and this is the responsive neurostimulator, which has an ability to detect seizures from electrodes that are implanted in the brain. These are also patients with Lennox-Gastaut. In this trial, there are electrodes implanted in, again, both centromedian nuclei, and there are also electrodes implanted usually over the prefrontal cortex bilaterally. There are two of these in each of the patients, one on each side with electrodes to the centromedian nucleus and then a frontal electrode. And then seizures are detected and then hopefully prevented by stimulation. So much more complicated. This approach is also being studied, and it's the same centromedian nucleus. Why? Because it's connected to a lot of things. Other pharmaceutical strategies, cannabinoids. So let me use this cartoon as a chance to say, clinicians love, and I will say require, a simplified cartoon to summarize the mechanism of action. Though the animations we heard were beautifully done, and clinicians are going to get lost in that. We need a cartoon to show us why does a drug work. So this is a cartoon trying to explain why cannabinoids might work for seizures. So you see numbers here, 1, 2, 3, and these are 3 places where cannabinoids might work. 2 of them affect intracellular calcium. The third affects extracellular adenosine. The final common pathway is decreased excitatory neurotransmission. A ridiculous oversimplification, but as you're explaining to patients what's novel about this, we need something that we can understand and then explain to patients. So, based on this mechanism of action, what clinical side effects would you expect? Assume it's going to work for some patients. It's not going to work for everyone. But what clinical side effects would you suspect? Well, based on the trials where you have clear side effect data compared to placebo, the clinical side effects of cannabinoids to date include, and these are common ones, decreased appetite, somnolence, and diarrhea. And the diarrhea may be related to the oil rather than the drug itself. So remember decreased appetite and somnolence. And somnolence is kind of a grab bag of somnolence, fatigue, tired. There's a group of things that it could be labeled. So effective drug, like everything, some side effects. Serotonin. Several compounds in development, or one approved already, fenfluramine, with mechanisms of action via serotonin pathways. Now, from a clinician point of view, serotonin and epilepsy, what does serotonin have to do with epilepsy? Well, it turns out that based on our very, very oversimplified understanding, serotonin modulates both GABA and glutamatergic transmission. All right, so a serotonin drug is going to work via GABA glutamate, hopefully resetting whatever is out of balance. So again, assume it's going to work for some, not for everybody. Side effects. What are the tolerability side effects of serotonin drugs? Well, to date, they have been decreased appetite, somnolence, and diarrhea that may not be related to an oil, but may be part of a serotonin mechanism of action. Effective drugs with some tolerability side effects. This is the cartoon that's used to describe the mechanism of action of fenfluramine, which works via serotonin receptors on the postsynaptic membrane to increase GABA, decrease glutamate, our old friends in epilepsy neurotransmission, but also has an intracellular effect through sigma-1 receptors, which I think clinicians are not used to thinking about. And again, we're going to see this cartoon and say, okay, all right, okay, GABA up, glutamate down, that's good. Tell me about anticipated side effects. And so we're back to somnolence, appetite effects, and perhaps diarrhea. So Dr. Hawkins described this slide as a very simplified description of the actions of CYP46A1 inhibition. So I chose this very simplified slide because this is what clinicians will want to see. Brain cholesterol, what does that have to do with epilepsy? And as Dr. Hawkins described better than I can, the hope is that CYP46A1 inhibition will lead to decreased excitotoxicity through the mechanisms that she explained quite eloquently. So you saw in the second animation the soticlestat phase 2 clinical trial results. Positive for Dravet, a trend, but not positive for Lennox-Gastaut. The side effect profile was some patients were somnolent, but the difference between the active drug and placebo was only 10%, which is pretty good. And then the other side effect that separated from placebo was constipation at a rate of 6% versus 0. So the intriguing thing, and I think a part of the discussion that I want to have, is as we learn more about brain cholesterol, what will this knowledge help us do to predict side effects in humans? Clinical side effects, because that's going to drive the boat of patient and caregiver acceptance of any novel therapy, whether it's DBS, a serotonin drug, a cholesterol drug, or something else. So here is just a reminder of the common adverse effects that we face every day. Dizziness, double vision, or classic sodium channel side effects. Sedation, cognitive impact, too much GABA, you're going to be tired, you're not going to think clearly. Alter glutamate AMPA, you can have behavioral instability, somnolence everywhere, and then appetite and the GI side effects, I think, have come almost to the forefront because some of our new drugs have unpredictable, sometimes favorable, sometimes not favorable impact on appetite, diarrhea, or constipation. So we're going to have to sort out all of those as we sort out clinical use. So back to my granted superficial addressing of precision therapeutics, genetic epilepsies, fabulous advances in the understanding. Treatment pathways, as you know, if you've been around the meeting, there are many, many strategies. The key is, I think, two. One, timing, how early, and how do you design the clinical trial? Because it is, there's a standard template for an anti-seizure medicine drug trial. There is not a standard template for a gene therapy or RNA or DNA modification therapy trial. So we have to figure that out, especially if we want to measure at a regulatory standard improved development or cognition without an endlessly long control group. All right, so that is it. So there are lots of novel pathways that have emerged. This is a very exciting time. And these novel pathways, cholesterol and otherwise, have lots of potential for improved efficacy in treating DEEs. But as a clinician, I will tell you that minimizing side effects is going to be extremely important. And the more we can predict, identify, and minimize tolerability side effects, the better position we'll all be in, and the better position our patients will be in. All right, so now it is time for the post-test. And, there are no consequences to getting a low score. As these questions were going up, I reminded at the medical school where I teach, the students will often say, when you give us an audience response question, please give us a choice for I don't know, because they hate to guess wrong. If they don't know the answer, they don't want to just guess. So if you don't know, just pick the best answer. So, all right. So, which type of brain cell performs the majority of cholesterol turnover and when impaired is hypothesized to lead to neurological disorders? All right. You've seen these before, so I'm not going to read the answers again. All right. All right. So, survey says astrocytes. This time you do get correct answers. Neurons are the correct answer. And we have ample opportunity to discuss why that is with our expert panelists. I'm not going to be able to tell you why that was right and wrong, but my colleagues will. All right, next. During cholesterol metabolism in the brain, which of the following biomarkers is the primary metabolite that can be measured in humans using plasma or CSF? You see the choices there. All right, survey says 24-OHC. I think I remember this one. There you go, correct. Very good. Next, which of the following is an advantage of preclinical models of DEEs? All right. So conditional or developmental expression of the trans gene is the most common response, and that is the correct answer. Well done. Next, blocking brain cholesterol catabolism in an Alzheimer mouse model reduced excitability and release of which neurotransmitter? All right, survey says glutamate, and that is the correct answer. Number 5, inhibition of which enzyme with soticlestat is thought to improve seizure outcomes? All right, survey says CYP46A1, and that is correct. All right, post-test question 6. Sustained status epilepticus has which of the following effects on cholesterol metabolism? All right, increase in 24-OHC levels is the majority, and that is correct. All right, great. You guys did very well. All right, so next, we're going to discuss some things that we didn't have time to cover, and we welcome your participation in this discussion. And then there'll be also dedicated time for questions, but feel free to participate in our panel discussion if you like. So first question I have is, how well do preclinical models predict tolerability side effects in humans, if at all?
Yeah, I mean, I think that's always the question that comes up when I talk about the preclinical models of seizure and epilepsy that we use. And I think it's important to consider that even though the maximal electroshock, the sub-QPTZ test, those models are typically associated with being predictive of clinical efficacy, they don't carry the same potential to show adverse side effect liability. And a lot of that evidence has come from research with animal models that have chronic seizures, where we have seen adverse side effects present more frequently than in the animal models that are not associated with chronic seizures. For example, and we talked about this a little bit when we were workshopping for this conference. In the early 90s, there was an NMDA receptor antagonist that was being developed for seizure disorders. And when it was trialed in healthy volunteers, it didn't show any adverse effects liability. However, when that compound went into patients with epilepsy, there were a lot of adverse side effects that came up and poor tolerability, largely because those individuals, the people with epilepsy, had higher brain and plasma concentrations than the studies up until it had predicted. And what was cool about that work, unfortunately for the compound, but what's interesting and helpful to really transform preclinical research, is that investigators went back and said, how can we have predicted that better? And they went and used animals that have chronic seizures, kindling models, and showed that kindled mouse models, which are animals that have chronic seizures, do show higher sensitivity and worsened effects with that investigational drug than the wild type animals. And so it's not that we can say, are we going to be able to see the perfect translation in our animal models for, say, cognitive side effects that we would hope to be able to predict in human patients. But what we can understand is that tolerability in using chronic seizure models, like the syndrome-specific models, the Dravet syndrome mouse models, at least gives us a better idea of potential for adverse side effects. I don't know if you want to suggest anything too.
Yeah, no, I think you
Okay, covered it.
So we see that, I think, in humans as well. The healthy volunteer side effects may be different from the side effects in patients with epilepsy. So that seems like that's a helpful parallel. I have one other question for my colleagues. And as I was following the slides about brain cholesterol and effects of an epilepsy and then other neurological conditions. So would soticlestat or drugs with a similar mechanism of action be expected to have any long-term impact on memory in adults that wouldn't be picked up in a short-term clinical trial? But based on the mechanism of soticlestat in brain cholesterol.
You're more of
A memory person?
Yeah.
Yeah, I mean, I think that's hard because assessing memory in animals is not always perfectly correlated with the human studies. And so, modulating cholesterol catabolism, based off of the simplified synaptic plasticity diagram, we do expect some improvements in cognitive function, but without really diving deep into the preclinical models to ask that question, it's hard to say whether or not this will lead to any improvements in cognitive function in people who have epilepsy or who have cognitive deficits to begin with. So that's a long-winded way of saying I'm not really sure,
Okay.
but I'm hopeful that we could ask that question in some of our model systems.
Thank you. So what questions do you have for the rest of us here?
I mean, Nicole and I can sit here and debate this question a lot, but how do you see the role of these syndrome-specific models in preclinical discovery if they are characterized by such high mortality and experimental limitations? How do we use that to work together and advance new therapies?
Right. So we made this mouse a long time ago and have, treated it with several different standard ASMs and novel compounds. When you have used it, for a very long time, you kind of know what to look for, what you're expecting. The high mortality, like when I think of Dravet syndrome, these patients themselves are fragile, and our mouse model is fragile. So much of what we see in our mouse model is what our patients have. So we see this induction of seizures based on increased core body temperature. We see spontaneous seizures with or without that initial seizure presentation when we heat them up. And then also, as we know, Dravet patients have this increased risk for SUDEP. So while my mouse model maybe has a little bit higher mortality rate, I think it's really important that we are looking at the mechanism of lethality with Dravet syndrome. And that was something that kind of stuck out to me with the MES data, that although it wasn't protective for that hind limb extension, it should be important to note that none of those mice died when they were treated with soticlestat, where a high percentage of the vehicle treated animals did. So there's multiple things at play, and especially with the Dravet mouse, they are much like the human patients that have Dravet because it's from birth and they're developing with, this whole system with, roughly 70 to 80% of the patients have SCN1A mutations. So that's what our mouse is, a heterozygous knockout. So I think it's really important to use like these models for the actual disease, because it instead of doing an induced method where the animal was able to grow normally for potentially a few weeks or a few months even, and then you give it its insult. So this is a true model of Dravet.
Yeah, I mean, I think they've really gained their position, and despite the fact that having the high mortality, they're starting to show their value.
Right.
So it's kind of an exciting time, at least from my perspective.
Yes.
Maybe.
And you know quickly if it works or not.
Yeah, exactly. I was also kind of curious, Dr. Dlugos, with the definition of LGS. I'm thoroughly confused about how it's defined, and I wonder, trying to think as a translational scientist and using the information at the bench to come back, or using the information from the bedside to come back to the bench, were there any signals or evidence that we could have predicted from the preclinical studies that maybe would have told us what to look for in the LGS models? Or is there something that you're seeing that you think is particularly interesting from that trial in the LGS patient population?
So I think I'll give a very old-fashioned answer. Short-term epilepsy trials for regulatory purposes are most efficiently done when patients have frequent, brief, self-limited seizures that are obvious. So a patient that can have 10 seizures in a week and then seizure-free for 4 months has unmet needs but is not a favorable clinical trial candidate. A patient that has a seizure every week that lasts 3 hours, that ends up in the ICU, has huge unmet needs but is not a favorable clinical trial candidate. Patients with LGS have way too many seizures, but they tend to be regular, self-limited, and obvious. So going back to the 90s, when felbamate was the first drug that gained an approval for Lennox-Gastaut, they leveraged those old-fashioned clinical realities of LGS and were the first to do a short-term clinical trial that led to an approval, and it's been a well-traveled path. That is about as unscientific an answer as you can imagine, but that's, from a clinical trial point of view, that's what LGS is. Frequent, regular, self-limited seizures that are studyable in a short-term trial with benefits to patients, just not transformative benefits. And the FDA, I think, is careful with they don't want to label something that is too broad, so the LGS criteria allows them to specify a label based on the clinical criteria of LGS. I'm not entirely sure that answered your question, but that's, I think, why we've had success clinically and regulatorily with LGS, despite a total lack of basic knowledge about what it's all about.
But it is characterized by slow spike wave seizures.
At some point.
Yes.
But the trick is, let's say a patient has an EEG report when they're 4 years of age that said they had slow spike and wave. It hasn't been seen since. And now they're in the trial as a 30-year-old. They're in the trial because of that EEG report from 27 years ago.
Yeah.
That's where the EEG, it's pathognomonic for the syndrome, but it falls flat when you really put it under the microscope.
Thanks.
So one of my questions is, we know that that soticlestat is being tested as an add-on therapy, much like many of the other new and novel ASMs on the market. Do you think there's a possibility that one day it could be used as a monotherapy?
Yes, absolutely. And in fact, based on recent approvals of other anti-seizure medicines, The approval for treatment of as adjunctive therapy in the treatment of XYZ has been replaced. The adjunctive has been dropped because anything that works as adjunctive therapy when tested worked in monotherapy. And it's a whole rough road to get to do a monotherapy clinical trial. So yes, I think it's a much smoother route to approval and clinical use of newer drugs in monotherapy, where they're typically better tolerated than as adjunctive therapy. So that's good news over the last, I don't know, 5, 6, 7 years. So we have a wide array of questions from the audience, and we can intermix your questions with yes.
Do you want to ask this one?
Mm-hmm. So you showed in your mouse model, so that looks great.
Mm-hmm.
The mouse model. So I have 2 questions for you. Number 1, have you seen that kind of data with other anti-seizure medications in your, particular model if you've tested?
Right.
That's number 1. And number 2, why do you think soticlestat works so well?
So we published in, I think, 2017. We put our model through like standard ASMs. We also did some polytherapy. We combined VPA with other approved drugs for Dravet. We had improvement in hypothermia for some, and as you would expect, lamotrigine, for example, we saw a significant increase in seizure frequency. I have tested a couple other novel compounds that we did see some benefit. I have never in my 15 years seen anything that had 100% survival with the mice. This was a very large scale study. We had 100 vehicle treated and then 100 of each. We had 2 different doses of soticlestat. So there were 300 mice in this study. And then we monitored them for 2 weeks. Continuous video monitoring to do all the seizures. And that was one of the questions I had that kind of goes with this, and that's why I wanted to answer. There was a question of whether it showed benefit in post-seizure recovery. So we also looked at the duration of each seizure. Very few mice, I think if I remember correctly, there were 4 out of the 200 that actually had seizures compared to 80 or 90% of my vehicle-treated mice that did. There wasn't a difference in duration of the seizures, but obviously the very few that there were there, we use a modified Racine scale, and all these seizures that we were having were so mild, there really wasn't a recovery period because it wasn't so intrusive. When we have some of these generalized tonic-clonic seizures in the Dravet mouse, they don't move for 30 or 45 minutes. Post-seizure. With the soticlestat, we would maybe have a little, we call rearing and paddling, so they go up on their back legs, and then they were fine eating, running around, acting like a normal mouse. So, that was very exciting. And then what was the second part? I'm sorry. Why do I think it works? It's a good question. We kind of go back and forth with this mouse model of talking about the SUDEP phenotype of, what is at play here. We know from all of our years of doing this is that it's not necessarily a product of seizure frequency that we see the death, it's the severity. So we had, and it's published in that soticlestat paper, we had several of these vehicle-treated mice that had over 100 seizures in that 2-week span with no death. And what was noted about those was that they didn't go into hind limb, which is a very severe end of a generalized tonic-clonic in the mouse world. So then we had others that had 1 generalized tonic-clonic seizure that went into hind limb and died. So, there's lots of research. Is it respiratory? Is it, what is it? So, quite honestly, I just don't know. I don't have an answer of why I think it worked. I just know it worked really well.
But I'll just chime in a little
Yeah.
bit on that comment too. And I think what Dr. Hawkins' data really points to, in culmination with a lot of the other things that I was trying to drive home, is that for a long time we have relied on kind of really standard screening models, maximal electroshock, sub-QPTZ, kindling, and then the Gehr's rats, the genetic absence epileptic rats of Strasbourg. Those are really the kind of traditional screening models. And the data that came out with soticlestat in those quote-unquote traditional models really points to the value of implementing these syndrome-specific models going forward in the 21st century. Like, these models are really challenging, really labor-intensive, they're super expensive, but at the end of the day, they probably are going to provide us with a lot better information about safety, tolerability, but also efficacy in a specific patient population. And whether those treatments can be, applied to the general population with epilepsy, that still remains to be determined. But at least for a very specific subset of individuals, these models are highly predictive, as we've seen with a couple other compounds. And I think that the information that you've provided in combination with the negative data in the traditional models, the data that we've collected in a viral infection model, the data that was collected in the MTLE model, it really points to a paradigm shift, if you will, in how we think about these models from a preclinical standpoint. And I will just leave it with, levetiracetam, or Keppra, came to market without any evidence of efficacy in that maximal electroshock or sub-QPTZ test. It worked in kindling models. It worked in a audiogenic seizure model. The 6 hertz, it wasn't actually where it was found. It went back later and showed that it worked in that model after the model was developed. But Keppra was really transformative for epilepsy. I would assume you would agree with that, Dr. Dlugos?
Oh, absolutely.
But perhaps we're at this kind of interesting juncture right now where, okay, we have a compound, we have these models that can differentiate and tell us about how we may be better able to treat epilepsy, because epilepsy is an umbrella term for a lot of different conditions. Maybe we need to start thinking about these precision medicine models.
Yeah. And I also think it's important to note too, like, when we did treat these Dravet mice, it was after seizure presentation. So we weren't doing this, very early on at, P0, P1. This was after we had that first seizure presentation. We started doing hypothermia to kind of baseline them, but then also the majority of Dravet patients who come in, that first presentation is a, fever-induced seizure. So when, we see just so many similarities with that model. So it's very helpful, I think, in this screening pipeline.
And sorry, continue on. It does have limitations. Everybody's Dravet syndrome model is a little bit different, but if you're consistently seeing activity across a range of investigative labs, that really points to the validity and the reproducibility and the rigor of the findings.
Correct.
So.
And that was one of the benefits too, because, as you've mentioned, there's several 1A mice. We were able to collaborate with Dr. Yamakawa in Japan, who has also a very well-known 1A mouse, and we demonstrated the same efficacy in the hypothermia studies. As you can imagine, we didn't redo the spontaneous because it takes a little bit of time, but.
All right, so we'll go through some of the questions you submitted, and feel free to add your own questions by standing up. So, question, so how well are the 6 DEEs managed with the current FDA-approved anti-seizure medicines? So the short answer is not well enough, but better than before those were approved. And a slightly longer answer is, in terms of seizure reduction, those treatments have gained approval based on anywhere from a 20% reduction in seizures compared to placebo to a 65% reduction in seizures compared to placebo in a 3-month trial. And those are good, but not well enough, because seizure burdens are high, so a 65% reduction is is very good, but it is not enough for the patients, and of course the side effects that we talked about. Next question, is there evidence that vagus nerve stimulation benefits Lennox-Gastaut patients compared with RNS or DBS? So thank you for asking this. So there is not a comparative trial of VNS versus DBS or VNS versus RNS, and it is likely there never will be. Certainly some of patients who receive a DBS or an RNS have had a VNS already that may have provided some benefit, but not enough. So yes, VNS is also part of the therapeutic strategy for DEEs. It doesn't have an approval for DEEs per se, but it is certainly used. And it's important to note. So thank you for that question, because VNS can be, it's minimally invasive and doesn't have drug-related side effects because it's not a drug, but it is not typically transformative. And the last question, and this is a great question, and if any product involving cholesterol metabolism goes forward, clinicians are going to ask this, and the question is, does cholesterol catabolism related to the ketogenic diet, how is cholesterol mechanisms of action with soticlestat, say, is it related to the ketogenic diet mechanism of action or any dietary therapy mechanism of action? I'm going to give what I think from here. I think they're separate pathways and they are not related. Is that correct?
Yep, correct. Okay.
Yeah. So parents and patients are going to ask this all the time. Oh, is this the keto diet in a pill we've heard about?
Right.
So it is. Sounds like it is not.
No, yeah.
You're gonna get back to the Dr. Hawkins lecture about how cholesterol can't cross the blood-brain barrier.
Yep.
So that's why you can't expect it to be that way.
Okay, so one of the questions I received is, what do we know about maturation of CYP46A1 enzymes in infants less than 2 years, especially less than 1 year? Again, if I go back to the mouse model, when these mice were evaluated with soticlestat, it is that kind of age range that we were looking at with the mice kind of coordinates with that age range in individuals. So when we did all of the plasma levels, we did see the reduction in 24-HC. So I think it would be safe to assume that, at that age, that there is enough expression of CH24H available to be effective in these younger patients. And as I mentioned too, we have basically complete expression of CH24H at about a year of age. So.
Yep. I had a question that was, how much improvement would you like to see in a preclinical DEE model before moving an investigational drug to a clinical trial? I think that's a great question and something I'm happy to discuss, partly because the idea that I've been kind of trying to present to you tonight is more that these DEE models are actually becoming really instrumental in us assessing the potential of a therapy for clinical investigation, especially if they show absence of activity in the more traditional mouse models, or maybe just not subpar activity. And so the DEE indication is particular to the clinical population or the model has to be particular to the indication that is being pursued. But I think, choosing the preclinical efficacy signals such as survival in a mouse model that's characterized by a high amount of mortality is a really robust efficacy signal. And for me, when I think about how I'm designing my preclinical investigations, I try to use those that understanding of what's happening in, say, Lennox-Gastaut patient population or a Dravet syndrome patient population to inform how I'm setting up my preclinical studies to be able to then see whether or not there is a signal. But if the signal that I see, positive or negative, is held up in like Nicole's lab, that's also really informative for me to at least be confident that this compound has potential moving forward. But at the end of the day, a mouse isn't a human, as much as we would like to model the human condition. And so we can't, the best population in which to assess efficacy is in the patient population. And hopefully the results play out in the patient setting as well.
So I would say again, in our mouse model, our first kind of pipeline that we use is we look at hypothermia. So do we see differences in that threshold to which they have seizures? We still, a lot of times, will continue on because what's really great about our mouse model is there's different aspects to looking at it. So not only can we look at seizure frequency, we can look at that severity. And then furthermore, while we do have a high mortality, as I mentioned earlier, it's a very quick readout, and it's not you can't really, it's dead. There's not a gray area. So, we have roughly 50% survival at about 4 weeks of age. So these studies actually can be quite high throughput with this model. I think for years there was kind of that negative, look at some of these genetic models that they were super, and there are components that are labor intensive, but if you can get a yes or no on survival, which then would be rated, we call it this SUDEP-like effect that we see. 4 weeks to see if that's effective is quick, in my opinion. And we can test things very quickly.
Oh, Ulrika?
I would like to add something, a small comment, because you showed so nice and brilliant work on animal research, which I think is really important, and I learned a lot more than I expected, to be honest. And I would like to emphasize that we all should also go out into the public and tell how important animal and in vivo research is, because we are facing it every day that outside our bubble, let's say, the people do not know what we are doing and why, and they don't understand. But it's so important for the patients that we do all these animal research, and I think we should more bring it out into the public how important it is, because especially, I don't know, in the US and Europe, it gets more difficult, and I think the people need more knowledge about the importance. But your work nicely shows how important it is and how it gets translated into the human, directly to the human patient. So,
Thank you.
Yeah. Cheers. Thank you.
Okay, so this is one. So please explain how CYP46A1 inhibition reduces neuroinflammation. So we kind of go back to this vicious cycle of hyperexcitability. Glutamate is glutamate activation. When we talked about having too much glutamate in the synapse, so it's just kind of this vicious cycle that goes around and around. So when we are blocking CYP46A1, therefore, down the line, we're reducing the amount of activity on that NMDA receptor. So therefore, in this feedback loop, then we could be preventing neuroinflammation. I'm not an immunologist, so.
Well, yeah, I mean, there's a separate type of neuroinflammation.
Right.
There's a couple different types of neuroinflammation too, and I think it comes back to the role of the EAAT2 transporter in astrocytes and how they respond to extracellular glutamate. They're responsible for glutamate clearing from the synapse. And so if you've got something that's interacting with that transporter that's residing on astrocytes and they no longer have the ability to clear glutamate from the synapse, well, then you're going to lead to excitotoxic damage in the brain, and that is going to initiate some neuroinflammatory signaling cascade that causes the neurological damage that we see preclinically in our basic science models that define models with spontaneous recurrent seizures. So I think it's, I think that kind of comes back to a question that we talked a little bit about too, with why maybe the traditional screening models didn't show any activity, is because more than likely those models, I mean, they're neurologically normal animals, seizure-naive animals, and so they don't have all the network remodeling and the excitotoxic damage, the changes in the glutamate load in the synapse. They don't have the same reactive astrogliosis that we know is associated with epilepsy. And so maybe you have to have a situation where you've got excess glutamate to lead to the hyperexcitable state that causes the neurological damage. And that's why compounds that maybe target cholesterol metabolism are more useful in models that have chronic seizures.
Okay, do you have concern with cholesterol accumulation in the brain with soticlestat? No. So the CYP46A1 homozygous knockout mouse did not show any behavior abnormalities. It had completely normal peripheral cholesterol levels. So for that particular, when that was knocked out, that was not of any concern.
I think our last question was kind of directed at the CYP46A pathway metabolism showed 85% of brain cholesterol was metabolized, but what about the remaining 15%? And I think there is some evidence about recycling of that remaining cholesterol level, and it kind of just resides in the lipid rafts of the brain, of the neurons. So it's more than likely just recycled.
All right. Other questions from the audience? All right. Let's see. So we did much of the homework already with doing the post tests already. So a reminder, the QR code gets you access to the program and what we call a clinical companion guide that in a chart form summarizes some of the classifications and definitions that we've talked about. I guess I'll leave that slide up for a bit. Thank you very much for your active participation. The questions, I think, were spot on, and I think the comment about the importance of animal research is huge. Not just the general population doesn't understand the importance of it. I think a lot of neurologists, even epileptologists, don't as well. So I think this program put together with a mixture of basic scientists, but with clinical information, I think we need more of these programs. And like I said at the beginning, I learned a lot. I hope you did as well. And you can complete the evaluation. And thanks very much. Enjoy the rest of your evening.